So we're in the Newport Aquaculture Lab at Hatfield Marine Science Center in Newport, Oregon, and this is the main wing of the building where we do most of our work. To our right here we have this room, which is set up for ocean acidification experiments. Here we have seawater systems. We can get both hot and cold seawater as well as dechlorinated fresh water coming out of the wall. We have head tanks where we can aerate our water overnight with soda lime, which helps to remove the carbon dioxide from the air, which then removes the carbonic acid from the water, which helps to raise the pH. We also have carbon dioxide cylinders for when we need to simulate high pH or low pH conditions with high levels of carbon dioxide. And right now we also have just a lot of different seeds and basic physical equipment that we use to try and basically run an oyster hatchery. This next room here is our Laurel hatchery room. Each of those 30-liter buckets often contains a single genetic cross or an oyster family. And so during the hatchery season, we have more than 100 of those buckets going in here. And it's a full-time job just to keep those going so that we can rear lots of different oyster families. We have a walk-in fridge, and then in the back of that, we have a -20 freezer. And this room here is where I spend most of my time. It's set up really nicely to do larval experiments with bacteria. We have lots of different room for setting up different size culture vessels. As you can see, we have one liter over here, we have 10 liters over here. The whole room is plumbed up to receive air from the main Hatfield compressor, as well as we have plumbing over there to receive air directly from the outside if we need a source of air with lower levels of carbon dioxide. We have head tanks so we can aerate water overnight to get it to the right temperature and pH. We have sinks, and we have this system here which plugs into a UV system so that we can sterilize all of our effluent water before releasing it back to the bay, so that we can work with different bacterial pathogens, mainly ones that harm oysters but wouldn't harm humans. All of those will get sterilized through the UV before we release that out. The next room down the hall here is our mini system, or our nursery room. This is where we take the larvae when they're a little bit older. After about 2.5 weeks, they'll move from the hatchery room over there to the nursery room, where each of them will be raised in these upwellers. There's normally water flowing in and out of these troughs. We add food directly to the system, and then as that flows up through, it passes through these upwellers and then passes out, so that the same water is never passed from more than one or through more than one family, and it's not recirculated through. It passes into the upweller and then out. The next room coming up is the cafeteria, or the algae production room. It definitely looks the most exciting of all of our rooms. We typically grow 3 different types of algae at once in these large cow walls, and then we have quite a few different species of algae that we grow on a much smaller scale, or we just maintain for later use if we end up needing different cultures. If one of them gets contaminated, we'll swap them out. So these are our holy stocks. We try and keep these really pure. We have a lot of different species of algae here. Then behind you there, we have our working stocks and carboids, which we can then use to inoculate these larger scale tanks if we need a higher volume effect. This is our chemistry lab. We have a lot of just basic equipment here, like an autoclave that we can use for sterilizing the seawater for algal production, as well as for preparing all the microbiology media. We have just basic things like a dishwasher, scales, stir plates, hot plates. We have a microwave for sterilizing glassware or melting agars, vacuum pump for preparing seawater filters to try and identify large quantities of bacteria, or even identify some viruses that are present in the seawater. Lots of pipettes, as well as just large amounts of bacteria or laboratory equipment such as glassware and beakers. Yeah, that's probably pretty much this room. Oh, we actually have a freeze dryer machine that we can use to freeze dry dulse or liposomes, which Matt Hoyer here does a lot of work with, and we're currently trying to freeze dry some of the probiotic bacteria for the project that I'm a part of, so that we can then have a shelf-stable product that can be sold to hatcheries or distributed to hatcheries in a very convenient manner. So the next room on my right is our Spat Lab, and we call it this because it has a large number of different microscopes that we can use for looking at the larvae in spat. So we have compound microscopes, we have a wide variety of inverted scopes as well as dissecting scopes and non-inverted microscopes. And we also have those hooked up to a camera that's hooked to the computer, so we can do a lot of image analysis of taking pictures of the larvae and measuring them and finding out what affects different growth rates of the larvae. We also over here have a laminar flow hood, which is really great for working with probiotic bacteria and various things where you have open cultures that you need to keep sterile. And then just to the left of that, we have a machine for helping to, it illuminates the background and provides a grid for counting different bacterial colonies on plates. We have a water bath, which is great for when you have molten agar that you need to keep liquid, as well as a spectrophotometer for identifying the density of your bacterial cultures. A centrifuge, which I use often for washing bacteria. I know it gets used for a lot of different components of this lab, as well as just a lot of storage space. We have more glassware and all of the basic materials of a lab. Over here we have a fume hood, which is used for all of our hazardous chemicals, as well as an incubator, which we use for growing the bacteria. A Coulter counter that's, it was initially intended to count blood cells, but we use it to count algae cells in seawater. So that's really handy when we have a flow-through system where we're adding the food in the beginning. We want to see how much is entering and then how much is exiting, so we can tell how much the bacteria or the larvae are eating. We have drying ovens. We have ultrasonic cleaners. Workshop area. We end up doing quite a bit of fabrication as we design various different experimental systems. There's always plumbing that goes into it. So a lot of that can be done here. We have a vise, we have saws, there's lots of PVC cement, as well as just an assortment of different tools that we need for, so pretty much whatever minor problems comes up, we can try and solve those. Anything larger, and we'll get the Hatfield facilities crew on board, and they're really good at what they do. This is we call this the Eukarya conditioning system because it draws water from and also drains to the Eukarya Bay. The water that it draws in here has gone through the Hatfield filtration system and is filtered down to 10 microns. So it gets out all of the copepods and other various critters that we don't want, so that we can try and control what's present in the seawater. But this is really good because we can have a continuous flow of water entering as well as exiting all of these tubs to try and maintain really good water quality. So we can bring the oysters in several months before we actually need to spawn them. We can raise the water temperature slowly to try and simulate summer conditions even in February to trick the oysters into getting ready to spawn so that we can then get them to spawn earlier in the year. So Hatfield has a large pump house where they do all of their filtration of their seawater. But we take it one step further because we need really clean seawater for what we do, and we use a huge amount. So behind me are our sand filters that we use to bring all of our water down to just an extra layer of filtration. And then after that, behind those, we have our bag filters here, which those are where we get it down to that 10 microns, the really fine level of filtration. In addition to oysters and some finfish research, we also do quite a bit of research with dulse. And these are our holy stock tanks where we keep different dulse varieties in a heavily concentrated manner. And then we can pull this out of here and use it to inoculate our larger dulse systems. And this is our algae production greenhouse. We go from the algae room, which I showed you earlier, those large cow wells, the cylindrical tanks, get pumped out here when we need an even larger volume. And we can combine different species of algae together into these tanks and then pump these back into our various flow-through systems to ensure the larvae are always getting a really nice, robust algal diet. And this is actually one of the things that sets our particular research group apart from a lot of other shellfish research groups is access to a facility like this. Having live microalgae can make a really big difference in the overall health and growth of larvae. We have just a lot of different tanks, so we can be growing and feeding multiple ones at once. We try and always feed them when they're at the optimal algal density, and we actually have a full-time algalogist who's in charge of taking care of and growing all of this algae. These are all experiments that are being done with the dulse. They're growing it under various conditions. I think in the past they've done different salinities, pH, temperatures, just to try and figure out the optimal growing conditions for it. And they've also found that by altering the number of nutrients that you put in, various trace metals, things of that sort, you can have different colored dulse, you can get different texture. With different aeration rates, you can get very different shaped dulse. low aeration, it grows totally different than it does under a high aeration cumble culture. And so we're trying to figure out, one, how it grows best, and two, what consumers want, so we can try and then tailor the growing conditions to find a product that will make consumers really happy and excited about it. So you can grow this super sustainable product and get people to eat it. Here we just have more seawater tanks. This whole side of the courtyard drains into a quarantine system, which then flows into that facility there and is completely sterilized before being released to the bay. So if we need to bring in animals from anywhere other than the Aquina Bay, we want to make sure that we're not spreading pathogens where they shouldn't be. So we keep all of that on this side of the courtyard. And I'll give you a quick peek into our, we call it the Chlorine Shack, where all of the sterilization goes for the quarantine side. So we have the capacity to UV the seawater if needed, but all of it goes in there and just slowly flows through that and gets chlorinated before it's then the chlorine neutralized and it's released to the bay. This is also our quarantine side. We are now in the disease wing of the Newport Aquaculture Lab. We share most of this facility with NOAA, who actually owns the building. Everything on the other side of the tarps belongs to NOAA, but this corner here is our quarantine conditioning system. It operates really similar to the Aquina conditioning system, except for instead of draining directly into the Aquina, it goes through that quarantine first. You can see we have water constantly flowing in and out of all of these tanks. What you have up top here is spat that are ready to be outplanted. They're now about the size of a nickel, and those will be put into cages and then put out into the bay. Down below here, I have broodstock that I'm conditioning to get ready to spawn for all of my experiments. That's another really great aspect of these facilities, is we're able to condition our own broodstock, which means I can control every variable of the spawn myself. I'm not relying on hatcheries spawning the larvae and then shipping them to us. So you take out any of the possible stress of that shipping period, and we have larvae on hand anytime we need them. So we're still in the disease wing of the Newport Hopkins Lab, and in this room we have a lot of -80 freezers, which are really good for storing probiotics over long periods of time. I know there's also some tissue samples in there for DNA analysis, but basically anything that really needs long-term cryopreservation, we have the ability to do that here with our -80 freezers.